next speaker is Wenming Huang, who will talk to us about stuff from Adam Antebi's lab.
Okay, great. Okay. Thanks, Morten. It's my great pleasure to be here. So I'm a postdoc from Dr. Adam Antebi's group from MPIH. So today I'm going to share a story how splicing factor regulates aging in C. elegans. So this work has been recently accepted by Nature Aging. But if you need more information about the detail, you can come to my poster 87 after the meeting. So splicing is one of the most important process for gene regulation. So it removes intron region and negates exon to form mature message RNA for translation. And in recent years, more and more studies reveal that the dysregulation of splicing might be one potential hallmark of aging. So for example, many splicing patterns, they change with aging process. And the expression of many splicing factors, which control the splicing pattern, also change with aging. Yesterday, I think Alex has also mentioned this in his wonderful talk. So what's more and more studies from model organisms also provide direct evidence that many splicing factors are directly associated with longevity. But compared with other hallmarks of aging, the splicing, the function of spliceosome and splicing in aging just studied. So many questions, there are many questions remain unclear. For example, there are more than 300 splicing factors in humans, but are they equally important for aging or longevity? Another thing is if there's any important splicing events which drive aging process. So in Adam's group, we initiate a genetic screening for lower longevity regulators with our favorite model, C. elegans. So here we use cold tolerance as a proxy for longevity. So basically, we isolate cold resistant mutants. So this mutant which can survive under the 230 degree. And then with the mutant, we perform Max Planck assay, and with the long-lived mutants, we perform genetic mapping to figure out what's the causative genes. And from the screening, we identified one long-lived mutant which carry a mutation with ARMY6. So this mutation changed glycine to aspartic acid in very conserved locus. ARMY6 encodes the ortholog of human PUF60, which is a component of spliceosome. It facilitates the 3' splicing site regulation during the early stage of splicing. And the mutation with PUF60 will cause the Hyde syndrome in the patient. And this syndrome is associated with very severe developmental defects, including short stature and intellectual disabilities. So we are very interested with this mutant, and we characterize it in detail. We found surprisingly this mutant is a partial loss function. So because overexpression of ARMY6 can fully rescue the cold tolerance phenotype and the left span phenotype, we found that this might be caused by the decreased protein level as revealed by Western blot. So in the ARMY6 mutant, the protein level for ARMY6 itself is decreased. So next, we're wondering if ARMY6 knocking down can mimic this mutant phenotype. So here is a very brief life history for the worms, and we're knocking down ARMY6 from different stage. And not surprisingly, if we're looking down the gene from egg on, the worms are short-lived because ARMY6 is essential. But when we're looking down ARMY6 right after developmental stage, which is day 1 adulthood, the worm is still short-lived. But surprisingly, when we're looking down the ARMY6 from day 4 adulthood on, which is almost the end of the reproduction period, the worm, they are long-lived, which mimic the ARMY6 mutant phenotype. This is very striking and very interesting result. If this holds true for humans, it means that we can start our longevity intervention even after 40 or 50 years old. So we found the ARMY6 deficiency increased lifespan. So what's the mechanism? So ARMY6 mutant is cold tolerance, and surprisingly, or not surprisingly, this mutant is also heat sensitive. So if we increase the temperature by 530 degree to 2530 degree, at which wild type worms can grow happily, the ARMY6 worms, they will become arrested, which you can tell from the very small body size of the worms. We don't know why, but this defect can be rescued by ARMY6 overexpression. So we are thinking maybe this provides a very good opportunity for us to perform a spread screening to figure out the downstream or the interactor of ARMY6. So we performed the genetic screening based on the ARMY6 mutant, and we isolate mutant we isolate mutationized worms which can develop again at 25 Celsius degree. And from the mutant, we identify the causative gene for two robust suppressors. So one mutation which we identified is ARMY6, which carry an intragenetic mutation with ARMY6. So this is the original mutation, and this is the new mutation, which suppresses the defect. We also identify another mutant with RBM39, which is also a splicing factor, and it has been shown to interact with POP16 in human cells to regulate the splicing process. And as expected, both suppressors also suppress ARMY6 longevity phenotypes. So it's striking that both suppressors, they are splicing factors. So we're wondering if there are any key splicing events which mediate this longevity process. So we perform on-seq on these two mutants, and we focus on the intron tension, because intron tension is one important mechanism for regulating gene expression, and normally is associated with decreased gene function. ARMY6 increase the intron tension in general, and we narrow down our candidates. We narrow down our candidates to the events which can be suppressed by both suppressors, and then we perform next-span screening to figure out to looking for the genes whose knockdown will mimic ARMY6 longevity phenotype. And from screening, we get a very promising candidate, ARDD8. So ARDD8 knocking down increased lifespan, but this longevity is not additive with ARMY6 mutants. We also confirmed this result by using another ARDD8 non-allele. And so ARDD8 encodes the phospholipase C beta 4 in humans. And this protein can be activated by G protein, and after activation, it will generate a second message, DAG and IP3, to regulate the neuron function as well as metabolisms. So from our on-seq, we found that the ARMY6 mutation increased the intron tension of ARDD8 in the intron 8, and RBM39 suppresses effect. We also found that the intron tension mainly happens in the neurons by performing the RPCR experiment with neuron-specific isoforms by using neuron-specific primers. So next, we asked if ARMY6 also changed the protein level for ARDD8. So to this end, we tag ARDD8, endogenous ARDD8 with a front protein. And as you can see from the pictures, so ARMY6 mutation will decrease the ARDD8 protein level, and this can be reversed by RBM39. This is the convocation. So we are thinking, so if the decreased ARDD8 protein level in neuron regulates longevity, this means that if we can restore the ARDD8 protein level in neuron, this will suppress ARMY6 lifespan phenotype. And that's exactly what we observed. Not only ARDD8, but also ARMY6 overexpression in the neuron tissues can rescue or can suppress ARMY6 longevity phenotypes. So this brings to another question. So what's the downstream signaling pathway mediating this ARMY6 ARDD8 longevity? We have performed many genetic epistatic analysis, and we found that the ARMY6 ARDD8 axis might interact with mTOR signaling. So because mTOR inhibition in worms increase lifespan, as here revealed by the REG1 non-allele, and we found that ARMY6 mutant does not further increase REG1 longevity. This is also the same case for ARDD8 knocking down. We also assessed the TOR activity by using T5 nuclearization in worms. And we can see here, upon ARMY6 mutant and the ARDD8 mutants, the nuclear localization of HH30 is also increased in both conditions, indicating that the TOR activity is down. So TOR is conserved, and ARMY6 is also very conserved across species. We are wondering, what about humans, if the regulation of TOR by ARMY6 is also conserved in humans. So for this one, I would like to thank our wonderful collaborator, Kostas, and his student, Stephanie. So they used the amino acid starvation and refeeding assay in HEC cells to assess the regulation of TOR signaling. So they performed experiments, and their results beautifully show that when knocking down POP60 by siRNA, the raptor expression level is down in both starvation and refeeding conditions. The raptor is one core component for mTOR complex. And what's more, the POP60 knocking down also suppress the reactivation of mTOR upon amino acid refeeding, as revealed by T5 phosphorylation as well as S6K phosphorylation. They further show that this decreased TOR activity is partially caused by the decreased a lysosome localization of mTOR, because mTOR needs to be transferred to the lysosome to be activated. And we believe that this might partially explain why those Weyhaight syndrome patients, they show delayed developmental defects. So as a brief summary, we have found from worms the ARMY6 mutation increased lifespan by regulating splicing and TOR activity. We also confirm in mammalian cells that POP6 inhibition also suppresses mTOR. So as this picture has been shown by Matt yesterday, I completely agree that. So regarding the aging mechanisms, we know so little about it. We just touched the tip of the iceberg. But I also believe that by performing unbiased screening using simple model organisms like C. elegans, it provides a wonderful tool to address these questions. We can know more about the mechanism of the aging. So with that, I would like to thanks everyone for listening, and thanks to the ARDD organizer. So special thanks to the people who contribute to this project. And in the end, I also want to take this opportunity to post a small advertisement for myself. So I'm going to finish my post training, and I'm looking for position right now. So if you have any, if you are looking, so if your company or if your group is looking for someone who working with aging or drug screening, please reach me. I'll be very happy. Thank you.
Great talk. Last question for Wenming. A question? We have no question.
When can you start?
When can you start?
Right after the meeting. Yeah, very soon. Yeah. Thanks.
And is the conservation between these processes and humans? For example, EGL8.
For this one, it's not. Unfortunately, it's not. We also test PRC before in human cells, and it's not. So okay. I think it might be mediated by something else. Yeah.
Okay. Okay. Well, super interesting talk. Thank you so much.